Proteins are the bricks and mortar of life, without which existence would crumble. Hello everyone and welcome back to AHS Guide. Today we are diving into amino acids and protein chemistry, our foundation stone of biochemistry and life sciences. By the end of this session, you'll have a clear picture of how proteins are built, classified and studied. And trust me, once you understand this, everything from enzymes to the hormones will start making sense. This video is designed to feel like a real interactive lecture, so keep your notebook handy, pause the video when needed and try to answer the questions in the comment section. So the first topic of today will be about the general structure of D and L amino acids, the proteins, the molecules of life. So proteins are the most abundant organic molecules in the living system, making up almost 50% of the cellular dry weight. They are the basis of structure and function in life. From enzymes to antibodies, from hemoglobin to keratin, everything is made up of proteins. So the fun fact here is that the Greek word proteos, meaning holding the first place, is from protein is derived. So the word proteos, which is taken from Greek, means that it is holding the first place. And yes, proteins are important and holds the first place in our nutrition and our body. It was first suggested by Berzelius, a Swedish chemist, who later introduced by Mulder in 1838, describing the proteins as the high molecular weight nitrogen-rich compounds in both plants and animals. So Berzelius suggested the name, it is a highly important organic compounds, but the name protein was first used by Mulder. So can you think of any one protein which you encounter in everyday life? Pause the video, think about it and put it into the comment section. Now the general structure of an amino acid. So here is the structure of a simple amino acids. Note that each amino acids forming a chain will form a protein as a whole. So we'll first see what a basic unit of a protein, which is amino acid, is like. So this is the general structure. You can see a central carbon followed by an amino group, which is NH2, a carboxylic group COOH, a hydrogen atom and a variable R group. So R group can be a chain of carbon or any of the functional group or any of the chains that can be added to the amino acid group. So the difference between D and L amino acids lies in the arrangement around the central C carbon. So proteins in living organisms are usually and mostly and exclusively made of L amino acids. So since we have seen the general formula of amino acid, wherein you have a central carbon atom and surrounding which is the amine group, a carboxylic group, a hydrogen atom and an R group. So this is how the general structure of a amino acid looks like. If it is in ion state, you can see there is exchange of the hydrogen between the amine group and carboxylic group. Whenever this state is there, it is said to be that the amine group is ready for a reaction. So it is an active state wherein it can take up a ion or a given ion. Now the optical isomers that you can see, which is L and D forms, we'll see what L and D forms are. There is an asymmetric carbon where a C is attached to, carbon atom is attached to 4 different groups. So these 4 different groups can be in different position, which will determine the optical isomerism. All the amino acids except glycine alone, all other amino acids have the optical isomers. Optical meaning illusional, optical meaning a mirror image of it can be seen. So isomers are similar carbon atoms with different arrangement of the other groups around the carbon atom. Now L and D form is based on the L and D glyceraldehyde configuration that you can see. Then proteins contain L alpha amino acids in them. Now here you see a D-glyceraldehyde and an L-glyceraldehyde, and you can see that the OH group is at different position, and when you place a mirror in between both the D-glyceraldehyde and L-glyceraldehyde, they form a mirror images, and hence they are called the optical isomers. Similarly, a D amino acid and a L amino acid, you can see there is a difference between the position of amine group in the amino acid group. Here NH2 in the D amino acid is on right hand side of the carbon, and in L amino acid it is on the left hand side of the carbon atom, forming a mirror image of each other, and that is why they are called D and L isomers. These D and L isomers are dependent upon the carbon atom attached to the amino group wherein the difference is observed. That is what is optical isomers. Now the elemental composition of the proteins. Proteins are mainly composed of carbon, hydrogen, oxygen, nitrogen and sulfur. So these are a different percentage that is present inside the protein. So carbon has the major percent of presence in the protein, which is 50 to 55%, and hydrogen has 6 to 7.3%, oxygen was 19 to 24%, nitrogen about 13 to 19%, and sulfur 0 to 4%. Sometimes the proteins might contain phosphorus, iron, copper, iodine, magnesium, manganese and zinc as its R group, or any other addition to the proteins. So the average nitrogen content that must be present in the protein is about 16%. The Jalda's method is used to estimate the percentage of nitrogen present in the protein. So it is a method that is used to estimate the amount of nitrogen that can be present in a protein content. Now the polymers of protein amino acids. Proteins are formed by polymerization of L-alpha amino acids. So a numerous number of L-alpha amino acids are bound to each other by polymerization method. Around 300 amino acids exist in nature, but only 20 standard amino acids are directly coded by our DNA and is used in the protein synthesis. So the analogy that you can remember will be dealt later, but first get to know that the standard amino acids that are present in human body are only 20%, 20 of them. So these 20 standard amino acids can be synthesized and coded by the DNA, controlled by the DNA. So the reason for only 20 standard amino acids because there is a universal genetic code that allows to code the protein to be synthesized by the DNA. Anything that is derived from these 20 amino acids is called a derivatives. So these 20 amino acids further hydrolyzed or chemically modification is done, then it is a derivative of these 20 standard amino acids. Now the next topic is structure of proteins. So how is a structure of proteins created? Proteins, as we saw, it is a polymers of L-alpha amino acids. That is, there are many L-alpha amino acids that are bonded to each other in the form of a polymer, and these polymers together form a protein. So there is a branch of amino acids or a chains of amino acids which together form a polymers, and these polymers together form a protein. It is a complex organization. A 4-structure level of protein is formed. So there are 4 structures for a protein to be formed: the primary structure, secondary structure, tertiary structure and quaternary structure. The primary structure consists of the linear sequence of amino acids. So there is one straight line of amino acids which form a sequence and are in the form of a line or a rope. The secondary structure is a local folding of pattern, that is one single chain that forms a folding over local folding inside forming a knot is called a secondary structure. Now a tertiary structure consists of a 3D shape of a single polypeptide. So there is a single polypeptide which forms a 3D shape. A quaternary structure is arrangement of multiple polypeptide chains together forming a quaternary structure. Now we'll see the image of each one of them. So here in the primary structure you can see a single straight line. So that is a linear sequence of amino acids that is being designed by the DNA. Now secondary structure is like a spiral. So it is forming a local linkage or local folding of the protein primary structure. Now tertiary structure is a 3D structure. Here you can see the total how the patterns are polymerized together to form a single polypeptide which has formed a structure. Now coming to quaternary structure, it is a group of different types of polypeptides. You can see there are pink and white structures. So these are different types of polypeptides which are grouped together to form a quaternary structure. So that is how the structure of protein is divided into 4 different types. Now how to understand the structures? There is one analogy. Imagine there is a building. So in the building, the one brick from the building is an amino acid. A wall from the building, one wall from a building is said to be the primary structure. Now the twists in the wall. So you see there are different walls that are forming one whole room, right? Don't include the roof, just include the walls. So these are the secondary structure. Now I include the mold also, the roof as well. So one whole roof along with the walls, doors, everything is going to be the tertiary structure. Now the quaternary structure is the whole building consisting of all living room, kitchen, bedrooms, or the whole building as such. So that is called a quaternary structure. This is how you are supposed to understand. Don't write all these things. You can just write analogy, how you can compare an structure of protein along with the building. Now proteins are made up of polypeptides which are like more than 50 amino acids coming together, then it is a protein. The polypeptide is a term that is used for large chains without a quaternary structure. So that is how a protein is formed. Now let us see primary structure in detail. So primary structure is a linear sequence of amino acid that is decided and dictated by the DNA. It determines the function and the properties of the protein, and the genetic diseases is resulted from the defects in the primary structure. So any defect in the primary structure of a protein, then the defect is seen as a genetic disease in the person. So what happens is DNA starts coding. Well, how is DNA produced by DNA going to work? So basically in the nucleus there is a nuclear material. So the nuclear material that is in the form of networks is going to be translated, then transcribed and decoded later. So we know that the method of formation of a messenger mRNA is translated, then it is sent into the cytoplasm where it is transcribed by the ribosomes. So ribosomes take up the mRNA and along with tRNA they start coding the protein that is being commanded by the DNA. Once a protein is being synthesized, it forms the primary structure. Now the peptide bond is involved in the formation of the peptide long chains or the primary structure of the protein. So how is it formed? The formation involves the amino group, the carboxylic group, a peptide bond and a water molecule. So all together combine to form a peptide bond between amino group and the carboxylic group along with the carbon, central carbon. This peptide bond is a strong covalent linkage and it determines the rigidity and the planar structure, which is usually trans configuration is observed. So it's not like a cis configuration, it is a trans configuration. Then N terminal and a C terminal is written, that is polar participates in hydrogen bonding. So we have seen in previous diagram over here. So yeah, here we have the polar structures over here. It exists as an ion, right? So there is a polar structure formed because of the hydrogen that is moving and forming an polar structure in the peptide bond. Now the conventions that are used to determine the primary structure is while writing a sequence of N terminal and the C terminal and using a 3-letter or a 1-letter amino acid code for a single amino acid, and sometimes naming is done with the suffixes like ein, ein and ate with il also, and except C terminal amino acid all others are used to name them. So convention is a method of naming the amino acid group or the primary structure of the amino acid that is being depicted. So usually the sequence is to start from N terminal to the C terminal. You can use 3-letter or 1-letter amino acid code to name the amino acid group, and naming can be done with the suffixes like ein, ein, ate and a C-terminal amino acid is exception for naming. Like the suffixes are not used for the C-terminals. Even a single amino acid change can lead to cause. So that is very much important that you have to note. So here are a few images of whatever I explained right now. So there is a formation of peptide bond. There is one amino acid and another amino acid which has a C terminal and an N terminal. Together it is bonded by the hydrogen that is removed as a water molecule outside, and a dipeptide is formed. So there is a carboxylic group and amino group that is linked together to form a dipeptide or a peptide bond. So here the hydrogen that you see on both the sides is distributed and removed out as a water group. Now here there is another image of amino acid in a peptide chain, wherein glutamate, cysteine and glycine is the 3 amino acids that are linked together. There is N terminal and C terminal. So how do you name this? Here you can name each amino acid as E, C, G as single letters or 1-letter symbols for the amino acid chain or the peptide chain. Then otherwise you can use a 3-letter code for the amino acid. For glutamate it is glu, and cysteine cis and glycine gly. So this is a 3-letter convention that can be used. Otherwise you can write using the suffixes as glutamyl, cysteinyl, glycine. So you are not going to change the C terminal suffix. So it is going to remain the same. So same thing will be derived down. Now here we have the structure of an primary protein that primary structure of the protein. Here there is a trans configuration wherein the C terminals you can see here and here, they are at the opposite sides of the carbon chain, and hence it is a trans structure. Whenever there is opposite side, whenever the carbon has the opposite sides of groups, then it is called a trans structures. So this is the structure of a primary protein. Now, how to determine a primary structure? So, based upon the amino acid composition, hydrolysis and chromatography. So here the acid, alkali or enzymes using the chromatography technique you can determine hydrolysis and quantitative analysis can be used to determine the amino acid. What type of amino acid is present can be determined by the chromatography technique. Now the other technique used is the fragmentation. Here the non-covalent bonds that is present in the amino acid is broken down and urea or guanidine is obtained as a product. So then the the structure is said to be primary structure. So simple breaking of non-covalent bonds will provide with urea or a guanidine as the byproducts. If there is a disulfide bond, cleavage can also be done by putting a performic acid into the amino acid will give you a disulfide bond cleavage which determines the protein is a primary structure. You can use enzymatic cleavage with trypsin, chymotrypsin or pepsin elastase to break down the protein and determine the primary structure. Chemical cleavage wherein cyanogen bromide is added to the protein of primary structure and can determine the primary structure. Now, how is the primary structure sequenced? So, how do you determine what are the amino acids present in the primary structure of a protein? There are various methods. One such method is a Sanger's reagent, FDNB. Here, the N-terminal is determined using the Sanger's reagent. So, the Sanger's reagent is a reagent that is used to determine the N-terminal, which type of amino acid is present. Now, Edmonds degradation is also a method to sequence the N-terminal analysis with PTH derivatives. Sequentar is an automated sequencing wherein it is a machine or a technology that is used to sequence the primary structure of a protein. Overlapping peptides can reconstruct a full sequence. So if the peptides are overlapping on each other, a reconstruction of the full sequence can be done. Reverse sequencing from DNA does not show a disulfide bond or a post-translation modification. So reverse sequencing from DNA is that you take the primary structure of protein, take a copy of, translate it on a mRNA, and again you're going to sequence the mRNA. After sequencing this mRNA, you find out what are the reverse sequence of that protein that is being taken from the mRNA. So that's how reverse sequencing is done. Here is a Sanger's reagent. Sanger's reagent is added to the free amino acid or the protein that is labeled. It is hydrolyzed and the free amino acid is removed. A DNP amino acid is obtained. It is identified by chromatography technique. Now Edmonds reagent, similarly, protein is labeled with Edmonds reagent on, that is mixing Edmonds reagent with the protein that you have obtained. Once you hydrolyze with Edmonds reagent, you obtain a polypeptide with N-terminal amino acid. This polypeptide determines that it has a single structure or a primary structure of protein. The product that you obtained after putting an Edmonds reagent is the phenolphthalein hydantoin amino acid, that is PTH. You can determine PTH by chromatography technique. The secondary structure is the local folding of the polypeptide chain. It is done stabilized by the hydrogen bonds between the nearby amino acids. So here a pink color box kind of thing is one amino acid which is linked to the role of another amino acid. So you see that one single line that is being spiraled one above the other. So the bond that is formed between the two spirals is called the hydrogen bond. This hydrogen bond is going to determine the secondary structure. Now there are different types of secondary structure. One is alpha helix, beta plated sheets or beta benzer and irregular structures. In alpha helix, which was introduced or discovered by Pauling and Corey in 1951, they determined the alpha helix protein structure based upon the tight right-handed coil and R groups that are projected outwards. So that is how it was determined. Then a stabilized hydrogen bond between the amine group and the carboxylic group was seen, and there was around 3.6 residues or turns, that is there is 0.54 nanometer pitch, that is the distance between the two amine group and the carboxylic group was around 0.54 nanometer of pitch, or 3.6 residues or turns that is observed. Then there is a proline and cluster of acidic or basic residues that disrupt the alpha helices. Sometimes there are acidic and basic residues that are formed during this structure formation, which can destroy or which can stop the alpha helix structure formation. Now there is second type of secondary structure, which is beta pleated sheets. These are extended chains aligned parallel or anti-parallel. These have a hydrogen bond between the chains and may form from the separate chains or folded segments of the same chains. So here you can see the parallel structures, that is the A diagram is the hydrogen bond that is formed between the polypeptide chains. So there is one chain above here and the one chain below here. Between the two chains there is hydrogen bond formed. Here it can be parallel or anti-parallel. Parallel in the sense the chains are of same thing, that is same moving in same direction, and the bond is formed in the slanting manner. That pink line that you see is the hydrogen bond. Now N terminal, C terminal are in opposite directions, and this is the anti-parallel beta plated beta sheets that are formed. So this is how beta pleated sheets are formed, both parallel and anti-parallel, as shown with the diagram. So there is a difference between alpha helix. So helix is a spring-like structure. Beta pleated sheets are like the plain lines that run parallel or anti-parallel to each other. Now the other type is the beta bends or non-repetitive structure. Here you can see the beta bends, that is more repetitive structures are observed, and the structure of the secondary protein is different. It is bent at certain regions. So the diagrammatic representation of both alpha helix and alpha pleated sheets can be seen over here. So the spiral thing that you see is a alpha helix structure. The pink thing that you see is a beta pleated sheet. Now, tertiary structure, it is a fully 3D folding of entire polypeptide, which is a single polypeptide. There is a hydrophobic side chain inwards and a hydrophilic outward chains. The stabilizing force here, it involves hydrogen bond, disulfide bonds, ionic bonds, hydrophobic interactions, and van der Waals force. In primary structure, we didn't see any such stabilizing force because it was a single straight line of protein, amino acids that form a primary structure of proteins. In secondary structure, we saw only hydrogen bonds which stabilized the structure. But in tertiary structure, we see 5 different types of bonds that are stabilizing the tertiary structure of protein, which involves hydrogen bond, disulfide bonds or cysteine formation, ionic bonds, which is electrostatic, hydrophobic interactions, which is they are water heating interactions, and van der Waals forces that is also observed in these tertiary structures. The domain here is that it is a functional and structural unit. There are around 200 amino acid polypeptides, and usually more than 2 domains can be observed. Quaternary structure. So you can see the different bond formations. We'll see each one later. Now the definition of a quaternary structure is it is an arrangement of multiple polypeptide subunits. Previously in tertiary structure there is a single type of polypeptide unit, but here it consists of a multiple polypeptide subunits or oligomers. That was a This is a polypeptide with single type, and here polypeptide with multiple subunits, which is oligomers. And the subunits can be monomers, a single type, or a protomers, which can be more than one type. Dimers, there are two subunits. Tetramers with four subunits. So these are the structures that are formed in between two different chains of a polypeptide. Now the bonds that are formed here is non-covalent, which is hydrogen, ionic, hydrophobic interactions. Some examples of quaternary structure is hemoglobin, aspartate transcarbamylase, and lactate dehydrogenase. These are different types of proteins that are observed inside human body. So hemoglobin is present in red blood cells, aspartate transcarbamylase are again present in the blood, lactate dehydrogenase is present in the muscles. Whenever the perfect oxygen supply is not provided for the muscles to break down the glucose. Now there is a cysteine bond formation and there is a hydrogen bond formation between two different amino polypeptide chains. There is isoleucine and leucine formation, aspartate and lysine that are bond formation. So this is how two different polypeptide chains that come together and form the bonds, leading to formation of the different types of bonds and structures of proteins. The bonds in the protein are usually in two different types. One is covalent and the non-covalent. The covalent part as the sharing of electrons takes place, but in non-covalent there is no sharing that is happening, but a complete transfer of an electron can happen. Now in covalent, the peptide bonds are formed, and peptide bonds are usually seen in the primary structures. That is, one single straight line of protein amino acids that are linked to each other is called a peptide bond and is a primary structure. Now disulfide bonds is formed between two sulfur or cysteine groups. So this disulfide bond is again a covalent bond where sharing of electrons takes place and the bond is strong compared to the non-covalent ones. In non-covalent ones, there is hydrogen bond, hydrophobic bond, electrostatic bond, and van der Waals bond forces. Hydrogen bond is formed between two hydrogens that is present in the protein structure. Here the hydrogen bond are numerous and they stabilize the helices or the spirals or the sheets of protein structures. So helices or sheets is where we saw the secondary structure. Now coming to hydrophobic bonds, it is formed between the non-polar side chains wherein the hydrogen is ready to move off. So hydrophobic meaning the hydrogen is scared of bonding. So here the hydrogen is removed and the non-polar side is created wherein the clusters of chains come together to form the bond. And electrostatic bonds are between the acidic and basic side of chains wherein the donation and acception of the electrons takes place. So here acidic and basic side chains come together by the electrostatic force and the electrostatic bond is formed. Van der Waals forces are the weak dipole interactions between the two atoms that are present in the proteins. Now there is an example of a protein for you to understand. So previously I asked you guys to put an example of protein that is present in one example of a protein. So I don't know how many of you put into comment section, but here is an example of a protein that is present in a human insulin. This is a protein which consists of two chains, one chain A and B. The human insulin A chain has N terminal with glycine and C terminal with ASN. So human insulin has two chains with A chain of N terminal glycine and C terminal asparagine, and the B chain has N terminal phenylalanine and C terminal alanine. So that these are the two chains A and B with N terminal and C terminal as mentioned, and this hence human insulin is synthesized by pre and pro insulin. The pro insulin is formed, then an insulin is formed. How is it linked to each other? The two chains, how are they linked to each other? They are linked by disulfide bond. The cysteine together bonds and forms a disulfides. So here is an example of how an human insulin look like. There is an A chain and a B chain. So there are 21 amino acids in the A chain and 30 amino acids in the B chain. So the N terminal is at 1 and the C terminal is at 21. So at the first position of amino acid there is glycine in A chain and a aspargine at the C terminal of A chain. In B chain, the first amino acid is a phenyl alanine, and the 30th amino acid present in the B chain is the alanine. So this is the representation of how a sulfur to sulfur bond or a disulfide bond is formed between the two cysteine groups. So the cysteine group sulfur bonds are formed at 6th and 11th amino acid, 7th and 7th amino acid of A chain and B chain, 20th and 19th position of A chain and B chain respectively. A disulfide bond is formed to keep the human insulin at linked together. So where is human insulin produced? So human insulin is produced in the pancreas. The cells islet of Langerhans are involved in the secretion of these insulins to the human body. So human insulin is mostly important to regulate the blood glucose level in the human. So if any defect in the human insulin, then the patient is having a genetic defect which involves a diabetic. It can be either hereditary, that is being passed on from the parent to the child, or it can be due to genetic defect leading to diabetes. Now, how do you study a protein? A protein can be studied by a by conducting certain tests on it. So how do you conduct the tests on the proteins? There is one method for secondary and tertiary protein structures, that is X-ray crystallography and NMR, to determine the 3D structure of the protein. The other way to study the protein is by purification, that is you can precipitate it out to obtain the proteins. Amino or sodium sulfate is added to the protein and salting out is done. That is by heating up the protein along with ammonium or sodium sulfate, you obtain certain proteins. That protein is tested for the presence of the protein. Now ultracentrifugation is you centrifuge the protein in a test tube at a high speed rotation, which will give you the protein as a layer. Now electrophoresis is another method wherein you put the protein onto the isoelectric focusing, wherein the electrodes differentiate the different proteins based upon the chain or the weight of the proteins. Now ion exchange chromatography is another method. Gel filtration is also a method used to study the protein. HPLC is also another method to determine the protein. PLC is the high performance liquid chromatography, wherein high pressure, it also sometimes is called high pressure liquid chromatography. It is a test that is used in biochemistry, usually to analyze the proteins or any other metabolites that is present. Now the properties of protein. Here we have various properties that are specified, which we will be studying one by one. The first property is solubility. Now solubility is how the proteins dissolve in water or organic material. Proteins form colloid solutions in water, which is not a true solution, wherein they are not able to completely dissolve in water and form the clumps inside the water. So these are not true solutions. Now, due to their large molecular size, the solubility is low for a protein molecule. Now the molecular weight of a protein, it depends upon the number of amino acids that is present in the protein. Now each amino acid is approximately around 110 Dalton, and the protein might have up at a range of 4,000 to 4,40,000 Dalton of weight, molecular weight. Now, for example, some examples are being given. Insulin is around 5,700 molecular weight of Dalton represented in Dalton. Myoglobin is a protein present in the muscles, and it is 17,000 Dalton. Hemoglobin is a protein present in red blood cells, which is around 64,450 Daltons. Serum albumin is again 69,000 Dalton. So these are all the proteins that are present in human. Now the shape of protein, it can depend and vary in varieties, which is globular for insulin. So they are globular, and there are usually droplets of it. Insulin is in the globular shape. Oval shape for albumin. Fibrous or elongated in case of fibrinogen. Fibrinogen is a protein that is involved in formation of fibrin in blood clot mechanism. Now, isoelectric pH or pI, it is determined by the ionizable group of amino acids. So, at pI, the proteins are Zwitter ions. That is, when a protein is at a ionizing stage, wherein it can be neutral, or no migration of electric field is happening. No migration is happening in an electric field. When an amino acid is not ready to donate, it as a Zwitter ion state. Now the properties of isoelectric pH of protein is, it is minimum. It shows. So isoelectric pH shows certain properties like minimum solubility, maximum precipitation, and least buffering capacity, wherein the protein, whenever dissolved, shows the minimum solubility. That is, it does not solubilize completely, and there is maximum precipitate that is present. So whenever you try to dissolve a protein in certain alkaline medium or an acidic medium, it shows a precipitate. There is a formation of clumps, or the liquid is not completely solubilized with the protein. Now there is a least buffering capacity, that is, the proteins do not bond or form a buffer that is helpful in the reaction process. Now the few examples for protein with certain isoelectric pH is, for pepsin it is 1.1, casein 4.6, human albumin is 4.7, hemoglobin is 6.7. So all these are acidic, and the lysozyme is 11, which is alkaline. Now how do you determine an acidic or a basic pH? By determining the ratio. Now if the ratio of four different proteins, when added up and when divided, they produce the amount, wherein if it is having a greater than 1, then it is the ratio which is having greater than 1, it is basic protein. If it is less than 1, it is an acidic protein. That is how you determine whether it is an acidic or a basic protein. The precipitation of protein. So whenever a protein is tried to be dissolved or dissolved, it does not dissolve completely due to dehydration or because of the neutralization of the polar groups. Now how is the precipitate of protein formed at different reactions? If isoelectric pH shows less solubility, the precipitate that is formed can be determined here. So if the isoelectric pH is can also determine the solubility. Now if casein has a pH of 4.6, there is formation of precipitate and the protein precipitate is obtained. Now the other method for precipitation of protein is salting out by neutral salts. If added to the protein, it removes water and the protein aggregates are obtained, which is a precipitate of protein. If a protein has a high molecular weight, then the salt, the neutral salt that is being put can be reduced, or less salt can be put into the high molecular protein. Now this salting out method is used for separating the albumin and globulin. Now, salting in is another method of precipitation wherein a low concentration of salt is added, which increases the solubility of protein. Certain heavy metal salts like lead, mercury, and zinc at ionic states produce precipitates of protein in an basic medium. If acidic or an alkaloid reagents like TCA, tricitric acid, and picric acid or tannic acid, if added to the protein, they produce a protein anion complex, which is again a precipitate that is obtained after a reaction. Organic solvents can also be added like alcohol to dehydrate and take the precipitate protein. So this is another method of precipitation of protein. Now, color reactions are used to determine the presence of protein in a chemical or a precipitate. Now, how do you do it? There is a burette test wherein a purple color is with the copper ion in an alkaline medium is used to determine the peptide bond present in a sample that you take. Now, if it can be determined as positive if the reading is greater than or equal to 2 peptide bond presence or in the amino acids. So if the peptide has more than 3 amino acids, it is said to be proteins. So only amino acid that answers these tests is histidine. So histidine alone can be determined in the burette test. Now proteins is used. Some certain proteins can also be detected in biological fluids by the burette method, wherein a drop by drop chemical is added in the burette is added to the solution present in the conical flask, and rigorous shaking can provide a purple color solution in the conical flask. This purple color solution is an alkaline medium, and that alkaline medium might contain these chemicals present here. So there is a copper that is bonding between the N terminal of the 4 amino acid groups. This is a histidine group wherein a copper is forming a bond and producing a purple color to the solution. Here are 2 moles of urea that is at 180 degrees Celsius is present in the added to the burette to determine the presence of amino acids. So burette reaction is to determine the 2 peptide linkage formed between the amino acids. Similarly, there are different reactions that are done to obtain the specific number or group of amino acids present. Specific type of amino acid or a group of amino acid that is present. So burette reaction is for 2 peptide linkage between 2 amino acids. Then hydrogen reactions is used to determine the alpha amino acids that are present. Xanthoproteoic reaction for benzene ring of aromatic amino acids. So these are different types of amino acids, different types of groups. It might contain the any number of amino acids or the aromatic ring shaped amino acids, which we will be learning in the classification of amino acid. Just know the different reactions that can be used to determine the color. Use it to determine the presence of a specific kind of amino acid. So these are the color reactions. Now we'll see the denaturation of protein, which is another concept in protein. So denaturation, which means removing or the breaking down of the actual nature of the protein. That is denaturation of protein. So what is the definition of it? It is a disorganization of a native structure, which is if the protein is in secondary, tertiary or quaternary structure, now you break the structure and form a primary structure, which is basically intact. So you are removing or breaking down the structure of a complex one into a simpler primary structure. This process that is happening is denaturation. It leads to the loss of solubility. The protein loses its ability to get dissolved, and it also loses the ability to actively participate in the biological processes, or it can lose the ability to form a crystallization. It is like to form a crystals or the protein crystals. So all these abilities are lost when a denaturation happens. Now how is this denaturation going to happen? There are two agents of denaturation. One is physical, the other one is chemical. Now physical denaturation is by heating the protein. We are going to produce certain temperature at which the proteins break down into primary structures. Shaking. So by rigorously shaking the protein, it can also denature the protein. Ultraviolet radiation, that is sunlight, can also break down the protein, denaturate the protein. Then X-rays can also denature the protein. The chemical methods like adding acids, alkalis, alcohol, detergents, heavy metal salts, and urea can also disintegrate or disorient or disorganize the nature of the protein. Here is an image of how a native protein, the protein which is actually supposed to be like this, is being put in under a stress, a denature, and a denatured protein is obtained, which is a single strand without any bond formation between the two amine groups. So here you can see there is a bond formation. The dotted lines you see, that is a bond formation. Here there is no bond formation between two different groups that is present in an amino acid. Now, the characteristics of denatured proteins is they lose the native helical structure. The string structure or the helical structure they have formed previously is lost. It cannot be regained. There is increased viscosity and decreased surface tension. Because there is no surface tension, the viscosity is more. Surface tension is the attachment between the two different amino groups is decreased, and they do not have any pressure or tension that is causing the giving the shape to the protein. So there is surface tension loss and increased viscosity, so they are liquid like. So there is no proper shape to the protein. Becomes insoluble in organic solvents. So whenever the denatured protein is tried to be dissolved in the original solvent, it does not dissolve completely. That is insoluble. Easily digested. Previously, the bonds were very difficult to be broken down, and once it is denatured, it is easier to digest by the human body. So hence it is easily digested once the proteins are denatured. Usually irreversible. Example, cooked egg. So sometimes the protein that is present in egg, which is lecithin, once you heat it, it cannot be reversed back. That is the case in most of the proteins that are denatured. Sometimes at rare conditions it can be renatured and can be reversed. It is very rare. And coagulation flocculation is another property of a protein wherein the reversible and irreversible processes are observed. In case of irreversible denaturation, a coagulation leads to a semi-solid mass that is formed at an optimum isoelectric pH. That is, at a certain pH, the coagulation happens and a semi-solid mass is formed. Yes, one such example is heat coagulation test that is performed for a urinary albumin, wherein a sample of urine of a person is taken and heated and observed for any clumps in the urine sample. Once the urine sample has clumps, it is tested for the presence of urinary albumin in the patient, and the test is confirmed for any type of disease the patient is having. So that is how coagulation test is done, and the coagulation process is. Flocculation, here the precipitate of protein is formed at a certain pH, and this is a reversible process. Here the coagulum, the clump that is formed in the protein, it can be heated and be reconverted into the previous state it was. For example, casein precipitated in the milk. So the milk powder that you see is for previously in the solid state. So this solid state can be heated and reduced to a liquid state, and at a pH of 4.6. So you have to maintain the pH at the same level. Once the pH is kept at the same level, you can induce the reversible process. So that is how a coagulation and flocculation can be described for denatured proteins. Now that we have completed the topics under protein, which is the introduction of protein, from where the word protein is being derived, the structure of amino acid, which is L and D structures. Then we have seen the optical isomers. We have also seen the proteins, the properties of proteins, and the different structures of protein, the primary, secondary, tertiary, and the quaternary structures of protein, the bonds between them, how the bonds can be broken to form denatured proteins. We have seen coagulation and flocculation as well. So till now we have completed so many topics. The next topic, which is classification of amino acid and the peptide bonds and the functions of proteins and amino acid, will also be dealt. Till then, keep watching AHS Guide. If you have any doubt, please post it in the comment, as well as subscribe to this channel in order to get the update about different videos that I will be uploading. You'll also find the notes attached in the description with the link. Go press on the link to get the notes and download the notes as well. You